Great. Cool, thank you. Yeah, so I'm in Guy's lab, but still thank you very much to Guy, Jake, and Jenny for organising this and this opportunity. So I'm going to be presenting some work on a project that I did earlier this year, and this was largely in collaboration with the Alvarado Drug Discovery Institute over on the Biochemical campus here. And this project and the kind of the methods and the results, obviously, that I'll be showing revolve around using primary neuron-glia co-cultures using a sort of more up-to-date image analysis methods to use these in a high-content setting. So, what are we generally interested in? I mean, as you know from having heard Guy's talk on Monday, as a lab, we're interested in obviously microglia and more tending towards the sort of the side of the bad side of microglial inflammation. Obviously, what with the sort of the general consensus is that obviously microglia can get activated by a vast variety of things and damps or protein aggregates, etc., in a disease context in the brain, and many of the states, whatever they are, induced by those involve the production of things such as neurotoxic pro-inflammatory cytokines, reactive oxygen species, and also one of the focuses of our lab, this kind of excessive phagocytosis of synapses and neurons in the brain. So to kind of model these processes, particularly the contact-dependent processes, particularly being phagocytosis, you really need to be using neuron-glia co-cultures to sort of see that directly in vitro. However, despite we've seen plenty of talks that have used some of these cultures, they're fairly widespread, but in terms of their application in high content, high throughput biology, it remains rather limited in the academic literature, at least. And this derives from the kind of complexity, the cost, and it's just a bit of a limitation in the field of sort of drug discovery, target discovery at the moment, because obviously they'd be quite powerful when now we're sort of moving towards a focus on inflammation in these conditions. So one of the major challenges that is limiting this is, at least for imaging-based assays, is the analysis of these relatively complex systems. So what I'm presenting here is a sort of simplified schematic of the pipeline that I've come up with during the PhD. So what we're presenting on the top left there is an image of some of our cultures. So these are similar to the cerebellar cultures we've had mentioned already. So the neurons you're seeing are cerebellar granule neurons. And essentially these are primary tricultures from rats. Principally neurons, microglia, astrocytes. So you can see these images. So these are live cells stained with Herxt for DNA. NuO is a live neuronal dye. And then IB4 stains for microglial membranes, specifically in the brain. And what we can essentially do is use relatively recently developed open source tools to try and turn these images into sort of a quantitative output on neuronal loss, on microglial counts, etc. So in this case, the sort of key steps are essentially using CellPose, which some of you may know is a deep learning, open source, deep learning-based segmentation algorithm that is sort of basically nearly the gold standard in kind of segmentation at this point. And then once you develop the objects, so the kind of the shapes that you see on the right-hand side, then you can take all the measurements across your channels and then train a separate machine learning-based classifier to differentiate these. And while it might be obvious, the green cells are neurons, the red cells are microglia, and you can kind of set some thresholds, really classical image analysis. That proves quite challenging when it comes to relatively dense, complex cultures like this, where you have cells overlapping, and actually particularly for other cell types like the astrocytes, which you can see kind of in the background with relatively sort of large and diffusely stained nuclei, then you're really relying on relatively low signal-to-noise features, which proves challenging for a standard analysis. So what I'm presenting on the top right is a validation of this approach. So this is a confusion matrix. Essentially, how to interpret it is, so for example, top left, 95% of the neurons that are manually classified as neurons by myself are accurately predicted by the pipeline to become neurons, and then similarly for the other classes that we're differentiating here. hopefully that kind of validates the technique a little bit. We used this in a study earlier this year, which was published, and sort of slightly using this to introduce the concepts of LPS-induced neurodegeneration in these cultures, where we put this acute inflammatory stimulus on, so this sort of canonical TLR4 activator, and we get neuronal loss in these cultures, and much previous work has shown that in these cultures that is dependent on the microglial inflammatory reaction. And then importantly, in this paper, we showed that the SYK inhibitor and another one are protective against this loss in these cultures. So it's interesting to obviously compare that. We've had plenty of talks mentioning SYK and passing on TREM2, PLC gamma 2. Yeah, that can be, I'm not going to present any more data on that, but we can maybe talk about it afterwards. But this provided very good, sort of actually useful positive control for the screen that we ended up carrying out. So onto the screen. So this is now obviously fully in collaboration with the ADDI, and so they had the resources, the equipment, and the fantastic team there who helped me set this up. So essentially, one of the other very important things they had is an annotated library. So this is a library of compounds, many of which might be are in the clinic already, that have known on-target bioactivities. So this is important for going from a hit to a cellular target of interest when you're interested in the sort of cellular biology. So we selected a number of compounds from this library, just over a couple hundred, and then tested all of them single dose, 1 micromolar, triplicate plus or minus our inflammatory stimulus, with using sort of randomized plate layouts and so on to prevent any or limit any effects of sort of plate effects and stuff on the data, or edge effects. And the LPS was in this sort of checkerboard pattern again for the same reasons. And then we did this all 4 times, so separate preparations of primary co-cultures from separate litters of rats. So the controls, negative controls was DMSO, and then positive control, as I mentioned, was that Bay61 compound. So to give you an idea of what the kind of data looked like, the raw data, so the kind of obviously the individual points aren't important, but at this stage, but just to sort of say that we achieved a very significant neuronal loss with the LPS in this system. So in the absence of the LPS, we have a sort of relatively strong baseline, and then that's really significantly reduced in terms of the neuronal count per image. Obviously, we have some stuff going on, pushing neuronal numbers back up, which is obviously what we'll be discussing later. Similarly, with microglia, we see the expected increase, this microgliosis proliferative response with the LPS challenge. And then similarly, we get the data on other cell types. I'm only presenting some of that here, but just to show, for example, the astrocytes can be fairly variable, but don't change overtly with LPS consistently, at least. And also, this cell death that we're looking at in this screen isn't the nicest, cleanest cell death. You have a lot of necrotic and condensed cells that accumulate in the cultures as well. So in terms of the actual analysis on this data, so this is ordered by a kind of effect. This was all analyzed together, but for the clarity of sort of separated the LPS untreated and treated. So on the left-hand side, you're kind of looking at essentially neurotoxicity data. It's kind of the strength of that side of the assay. So reassuringly, for example, I've just pointed out a few interesting things here, but the most neurotoxic thing was rotenone, which is good because that's a neurodegenerative stimulus used for Parkinson's modeling. But also related to some of the DNA damage work we saw yesterday, we've got a ATM inhibitor, which is causing some level of neuronal loss, also a TDP1 inhibitor, which is also DNA damage related. Interestingly, TLR7, we had a talk earlier today on HIV, and vesatolimod is an HIV treatment, and caused some level of neuronal loss in this assay. Moving on to the kind of the key output of the study was this, obviously in the presence of LPS looking for hits that are protective, and actually we found a surprising number of hits that ended up just purely based on statistical significance. So again, highlighted a few. We've got another SYK inhibitor, which is nice, and compound 214, which is annotated there, is a very well-known TLR4 inhibitor, which is good because LPS directly activates TLR4, so it's nice to see that. And then the strongest hit, besides the positive control, was a glucocorticoid receptor agonist, which we'll kind of come on to that a little bit more, but it gives you a hint as to sort of we're starting to look at what pathways are involved in this degeneration using this screening approach. So this is obviously a lot. This is a list of hits ordered simply to show how we then sort of structured the focus for the, at least the discussion of the paper. And what this is kind of showing is all of these hits ordered by effect size, and you can sort of see the gene symbols of the targets that each hit hits according to the Kemble database. And where there's an asterisk, that means it's an agonist rather than an inhibitor, and then we can, in highlighted, we have things that are hit in that way, so agonism or inhibition, multiple times by the hit set of compounds, to try and draw out, perhaps the most interesting targets on which multiple compounds are converging. And things of note that we kind of pulled out of this, so we sort of classified some of them on the right-hand side, but compounds related to steroid signaling, adrenergic signaling, MAP kinase signaling were kind of particular targets. But it's certainly not to say that the sort of unlabeled or the non-duplicated mechanisms of action are not interesting. They are certainly worth a look as well. So to return back to some images, this just to kind of show what we're really looking at. So this is sort of similar images to before. We've also added in DRAC7, which is a far red live cell, live dead marker, and that allowed us to differentiate different types of obviously necrosis versus condensed cells. So this is LPS, completely untreated cultures, and this is kind of what they look like. And then you can see that this kind of really significant neuronal loss with the LPS treatment. And here, this is the TLR4 inhibitor. So we can see visually that obviously the analysis is working, and we're getting a protection of the neurons. What's interesting, and I want to kind of point out on this slide, is that this is just a randomly chosen other inhibitor, and while both are producing a nice neuroprotective effect, the other stuff in the culture looks completely different. So the microglia, for example, look completely different, and there are different numbers of them, etc. So it's kind of moving into that multicellular complexity that is hopefully one of the strengths of this type of analysis. So to focus on the microglia, we know that the microglia are important, as I've mentioned. So this is a plot plotting normalized neuronal counts versus normalized microglia. So you can see that we sort of start off up here in the absence of LPS. That's not even working, but on the left-hand side. And then we sort of move down to a high microglia, low neuron state, if you like. And then we're looking at the hits pushing back up towards the baseline. And again, labeled by these kind of classes. And some of the things to pick out, for example, is a number of the steroid signaling modulators in the hit compound set are actually increasing the number of microglia while being protective. Whereas, for example, some of the adrenergic receptor modulators in the hit set weren't really doing anything to microglial numbers. We can go a bit further on the microglia. We can go back into the data. I mean, compared to some of the morphology stuff we've seen, this is primitive. However, and in general, I think there's a very difficult to draw comparisons between 2D morphology and 3D stuff that we're used to hearing about. But we can go back in and we can see a shift with LPS, and whatever that represents in a kind of state sense, I don't think matters hugely. It's more the kind of that there is this shift, and we are seeing some changes. So, for example, a lot of the hit compounds are pushing the morphology back down towards that untreated level, even though they're producing a completely different sort of proliferation of the microglia. Finally, this is basically an integrative analysis of all of that data. So, essentially we've got kind of 16, just what we've extracted so far from the data. We have 16 kind of dimensions to play with. And this is kind of like the clustering stuff that we've seen, except it's just squishing it onto one dimension instead of two. So we've got our counts for the different cell types. We've got the morphology in there as well, in the absence and the presence of LPS. And then sort of trying to cluster and find hits that are doing similar things. And in a sense, this is now prioritizing and looking at hits. We're not screening, this isn't sort of looking for just neurons up or down. This is really looking for, if you like, a kind of phenotypic state of the culture as a whole, which is kind of a little bit beyond what most methods would allow you to do. So we see, first of all, just at the top, there's a kind of, the first kind of bracket is compounds which they don't seem particularly neuroprotective, though they are significantly so, and they don't really do much else to the other measurements that we make. But perhaps most interestingly, in the sort of central band here, we've got compounds which are actually quite strongly neuroprotective and actually do have clear effects on microglia. So again, a number of them are increasing the number of microglia. I should say that DMSO is here as a comparison. Also astrocytes, interestingly, and also completely changing microglial morphology, again, by the sort of blunt measure that we're using. So this implies that maybe we're achieving this, this stronger neuroprotective effect by actually messing around with glial states or glial proliferation. Finally, at the bottom, we've got a set of compounds which actually includes the positive control, which their phenotype, if you like, is actually quite a very strong reduction of glial numbers in most cases. And this corresponds with data that we have where depletion of microglia is protective because they are the ones mediating the damage. So I'd just like to then conclude, firstly, on the methods. So basically, this kind of study is aimed to be some sort of proof of concept that you can use relatively complex co-culture systems, even from primary cells in this case, to do some high-content screens. If you're interested in that sort of thing, from a high-throughput assay perspective, the Z factor was pretty good. Basically, anything from 0.5 to 1 is like a very, very good assay for high throughput, and this really sits in that range. But I kind of want to emphasize that this method is flexible. There are various limitations to the culture system we've used, and we've used small molecules. We've used LPS, and we've used primary cells. But in fact, you could make co-cultures very similarly from iPSCs, from cell lines. You could do a CRISPR screen. You could do all sorts of things. And actually, similar staining methods and similar analysis approaches would prove very flexible to that. And then this can support both high-content work and low throughput work, as we sort of found with our study on sick. And then finally, one of the key benefits is that you get this kind of really rich data set to look at this kind of like actual functional phenotypes in general in the cultures rather than just like a simple cell count for neurons, for example. And then finally, on the biology side of things, I didn't actually mention on the heat map slide, but you may have noticed that actually that central, quite interesting neuroprotective band, there was some kind of enrichment, if you like, for compounds affecting steroid hormone synthesis and signaling. So that means that glucocorticoid receptor agonist. There's also inhibitors, I think it was, of aromatase, so estrogen synthesis signaling. So some of that's quite interesting, obviously, with respect to some of the sex differences we've been talking about. I think that's a sort of perhaps the most interesting thing to come out of this data biologically. Plenty of other targets of interest, and there is existing literature, for example, on steroid signaling and glucocorticoid signaling being anti-inflammatory, similarly for adrenergic signaling of certain subtypes of adrenergic receptors. So there is literature supporting some of this stuff. And then, with that, I'd just like to thank my whole lab, particularly thank the team at the ADDI, particularly John Skidmore, John Clark, who's in the audience, and David Winpenny, who are just completely invaluable in setting this up. Henriette Willems is second author on the paper and produced the library a few years ago and advised on the compounds. And obviously I'd like to thank the DDI, and they also supported this financially as well. So thank you very much for listening. I'll just say that also all of the images will be on the Bioimage archive upon publication and accessible, much to the dismay of my home broadband provider, because it turns out uploading a terabyte of images is against some kind of fair use policy. But I don't know what that's about. And the code to replicate this stuff is on GitHub. So hopefully this is something of a resource. If you're interested in a particular drug or a particular target, it's possible to go into this data and look at images from co-cultures for that, and that might be useful for some of the people in this room as well, hopefully. So thank you very much. I'd love to take any questions.
Interesting talk. I'm also trying to attempt co-culture, and one of the common question I face from general discussions and from friends and all, how do you decide on the density, the ratio of neurons, microglia, and astrocyte? Because it varies with region, disease. So how do you decide on the density?
Yeah. So thankfully, that's something that I haven't, well, maybe not thankfully, but it's not something I've had to think about a huge amount with these cultures because we're not reconstituting from multiple cell types. So we don't actually have besides a sort of overt depletion of glia, for example, we don't have much control, although we do obviously aim to and do see consistent proportions of the cell types. But we don't have that kind of fine-tuned control. But what we do know is that this is kind of roughly representative of what you might expect to see in vivo in the cerebellum, for example. So that kind of supports that. But absolutely, to translate this into thinking about iPSCs or reconstituted systems, then that is really important. And I think it would be a matter of optimizing that such that it's physiological, but also such that it it is effective in the assay that you're working with as well. Yeah.
Thanks, Svetlana.
Great work, team. Thank you very much. A very naive question. So the sensitivity of different cell types, and I'm thinking about microglia, for example, astrocytes versus neurons, to a number of different compounds would be very different. So how do you settle on what concentration of a drug in a library to actually use? And how do you take into account these intrinsic differences that they can result in, various effects on cell fate decisions?
Yeah. So I think in terms of with respect to the obviously different effects on different cells, then that's kind of stuff that we hope to look at maybe retrospectively because we have the differentiation of the different cells in the analysis. So yeah, we're not really thinking about that on the compound selection stage. We chose 1 micromolar because if you think about it, actually, the more complex the processes that you're looking at, the more likely any given thing is going to hit something in that process, right? So we considered, and with the advice of the ADDI, considered, for example, 10 micromolar to be too high, and we just get all sorts of things going on. What I will say is that when we selected the compounds, we were selecting for compounds based on their potency on targets, right? So we were only using compounds that we knew, first of all, to be or according to their chemical properties, that were particularly cell permeable, and also only compounds which had on-target potency according to the chemical database that was below or more potent than 1 micromolar. So basically compounds that we know are doing something or should be doing something. But yeah, with respect to the cell type differences, then that's kind of maybe something to look at in the end data. Yeah.
Great. Thank you so much, Tim.
Thank you
Great
very much.
talk.